So, welcome students. We were in this course of biomolecular spectroscopy, we are dealing with UV visible spectroscopy at the moment, and in the last class I introduced you to Beer-Lambert's law. It is the absorption of a chromophore is quantitative because that depends upon the path length and concentration, and if we keep the path length constant, then absorption is directly proportional to the concentration of a molecule. Therefore, we can use this UV visible spectroscopy, the absorption concept, to determine the concentration of the molecule under investigation, and this is quite quantitative and often used for determining the concentration of a biomolecule. Now, then I moved into and discussed with you the chromophores that are present in a protein. So, if there is a cofactor like a heme that absorbs at a higher wavelength, like you see the blood is red color because of hemoglobin has a heme, or the protein has 3 intrinsic amino acid which are capable of absorbing light. This is tyrosine, tryptophan and phenylalanine, and we looked at the molar extension coefficient of tryptophan is much higher than tyrosine and higher than the phenylalanine. So, these are the 3 residue that absorbs. After that, and you can use this absorption of these residue to look at the conformational change that may happen in a protein. If it is buried, it will have less absorption compared to the when it is exposed. So, I showed you one example where helix is transitioning to a random coil. You can use this concept to understand the absorption concept to understand what kind of structural transition happening. Then I said not only amino acid, but also the bases absorbs light because they are basically they have unsaturated bond. So, if you look at all these bases A, T, G, C, all of these they are present, they have this unsaturated bond and they are capable of absorbing light, and they basically absorbs at a wavelength of 260, right. So, 260 for nucleic acid, 280 for protein, and you might have heard that when we want to check the purity of a sample, we take this ratio of 260 by 280 to see if your protein that you are purifying is contaminated with nucleic acid or not. We are going to discuss this concept in coming lectures, but that is the reason why you use 260 and 280 ratio, because 2A60 is the absorption happens due to this bases present in nucleic acid, and 280 our amino acids absorbs. So, if you look at the 4 bases in the water, the 4 bases that are present in a DNA, A, T, G, C and U also uracil, you look at their molar extension coefficients are there. So, here A G is higher, then A and T and U are little less compared to that. You look at the y axis that is changing, right. So, that is the absorption that is happening around here 260 for each of these bases, and that is the chromophore in nucleic acid. So, nucleic acid have a lower absorption than the sum of absorptions of individual nucleotide. This is simply because when they go for stacking, base stacking, the absorption goes down. So, the phenomena is called hyperchromicity due to the stacking or lining up the parallel plane of the bases. If they are like this, absorption will decrease, and when they open, it is more. So, bases will have more absorption if you just chew them up and just have in water, they will absorb more than when it is structured. So, that is called hyperchromicity, increase in the absorption. For example, native DNA displays significant hypochromicity, that means less absorption compared to DNA which is unwound, and absorption at 260 increases. So, this phenomena, this very nice phenomena can be used for understanding whether the DNA is melting or not melting. So, because of this base stacking, absorption decreases, right. So, in the structured DNA, this base stacking happens, absorption decreases, and when they basically they denature, it goes to the hyperchromosity, like denatured at 82, it becomes a single stranded. When it is denatured, that is a double stranded. So, absorption decreases, that is a hypochromosity. So, essentially this simple phenomena of UV visible spectroscopy can be used also to monitor the denaturation and renaturation of DNA, and that is what is one of the interesting phenomena that we use, structural transition that happens. So, DNA melting of two different DNA, you can get the what is the midpoint of denaturation. You can see this guy is going and melting like this, another is like this. So, stability you can Tm half you can measure for this one is around 50 and probably this one is around 40 or less than that. So, it says which DNA is more stable compared to the other one, and what we are measuring, absorption at 280 with respect to temperature, right. So, that is what one can do it in this phenomena of light absorption. Just again to remind you what we had in the instrument setup for measuring absorbance, what experiment we did, we had a light source, we had a monochromator, and this is the cuvette or sample holder, and this is a detector where we are detecting. So, instrument something looks like this, here is a detector, this is the simple sample holder, and this is the light source. So, all those experiments that we are doing of melting and hypochromosity and hyperchromosity can be done in this simple setup which is given here. It is not very costly, you require just light source which provides the monochromatic light UV visible, a monochromator, a sample holder which is here, and detector that basically detects the light, right. So, instrument what factors actually determines the accuracy of that, use of polychromatic light not ideal, you need to have a monochromator light, the stay light. So, it should be everything should be packaged in a box, that is what is requirement for this, otherwise we can just have on tabletop setup, that is not very efficient. So, you put that in a box, stray light should not come and instrumental noise should be minimized for precise measurement, and then calibration and regular maintenance is extremely important, although this comes about 10 lakhs rupees, but a normal one will come in 10 lakhs rupees, but if you want to go for a very precise, you have to spend little more time. So, calibration, maintenance, the lamp changing, the monochromator cleanness, and most importantly your cuvette cleanness. As a research scholar, you need to worry about cuvette which is clean, it should not be having sticky wall where something else will be sticking. So, all those are important consideration that we are doing, right. Now, source of error and deviation. If we are doing this experiment, first we need to know where we can make the mistakes. So, instrument deviation, non-monochromatic light or a stray radiation can cause the deviation. So, one has to see if our light is monochromatic light and there is no radiation that causes the deviation. We have to have this cuvette very well aligned, it should not be mismatching, the variation in the path length should not happen, and then instrument has to be calibrated well, right. So, whatever we measure is accurate, that is a calibration error, that has to be done very well. Now, there can be some artifact can be introduced because of chemical deviation. So, remember what we had discussed, A is proportional to the C and then we have a linear curve, but if the concentration is high, there is a possibility that molecular interaction, association, dissociation, reaction can happen, and that will change the property of the solution. That means, the sample under investigation is no longer behaving linearly, and therefore, this curve A versus C will no longer true hold. So, that is what is the, so up to some concentration we can measure it very accurately, but if concentration is increasing because of all these physiochemical change that may happen in solution, this can lead to chemical deviation. Now, how we can minimize the error? we need to use pure solvent, right, you should not be impure. Maintain consistent condition, right, temperature, the humidity, everything should be maintained. The most importantly, instruments should be calibrated regularly, and we have to operate in a linear range, which we explained here. Now, as we have given in 2 class back, extremely simple application is pulse oximeter, right, blood oxygen measurement, and that we can even analyze the environmental sample. Nowadays in winter, many cities in North India are populated. Simply one can use some of those UV visible kind of instrument even to measure what kind of like unsaturated carbon pollutants are there in the environment, and you can check the environmental sample measurement and how much concentration is there. That also, if you have some unsaturated carbon bonds floating in the air, you can find it out, right. So, to sum up what till now we have looked at, the Beer's law link the absorbance, concentration and path length. The extinction coefficient is a substance specific. So, it depends upon chromophore. So, substance specific measures the light of absorption. Spectrometry applies these principle for analysis, and calibration curve is key for finding unknown concentration. Deviation can impact the result, and application of this are diverse across the science and industry, all the way from pharma industry to biopharma industry to environmental industry. Even for like a forensic lab, you can use such environment, right. So, that is what is the summary of the absorption spectroscopy that we had. Now, just again to re-emphasize, since we are starting this course, let me re-emphasize few of those things that we have discussed in past, but again you get a very clear idea. I had discussed this electronic transition of formaldehyde. So, these are the transition that happens. These are the chromophore. Chromo means light and force means like a bearer, जो light को absorb करे, that is a chromophore. So, we can say from unsaturated electrons they can be transported to some non-bonding or anti-bonding. Because of that we absorb light, that is what we had discussed. And what are the factors that affects basically this kind of absorption. So, extent of conjugation is giving us chromophore. Like if you look at just an example, single bond, here we have a CC, the molecular orbital, I asked you to remember in the previous class, we have a highest occupied molecular orbital, HOMO and LUMO concept, right. So, HOMO to LUMO transition happens. So, if you look at double bond in ethylene versus conjugated system in butadiene, now that is the change in the energy that happens. So, conjugation, this refers to system of connected pi orbital with a delocalized electron in a molecule, which in general lowers the overall energy of a molecule and increases the stability, right. Alternative single and double bond like this, right. And impact of energy gap, that determines which absorbs where. So, and a simple isolated double bond like ethylene, here look at this, delta E between pi and pi orbital is large, and therefore, I explain this, that therefore, it requires high energy, high UV light and shorter wavelength for an absorption that happens around, say in this case 171 nanometer. And in case of conjugated systems, so if conjugation increases, the pi electron are spreaded over large region, that means delocalized. Similarly, in pi ring it is a delocalized, that this spatial expansion leads to a smaller energy gap, and therefore, a smaller energy gap, that means delta E has decreased. So, if delta E decrease, h mu that you need is lesser, mu lesser, frequency lesser, wavelength more, and that absorbs at a higher wavelength, right. Now, that leads to the factor conjugation and delocalization, and that leads to which is absorbing at higher wavelength and which is absorbing at lower wavelength. So, if the delta is larger, the lambda max is 171, means lower wavelength. If delta is smaller or moderate, your absorption happens at 277. So, for an example, lipofine, tomato, red tomato, it is red, its a delta E will be less, and that is for it absorbs at 500 nanometer. So, all those which has a delta E less absorbs at a higher wavelength, all those which have delta E more that will absorb in a lower wavelength like a blue and this will absorb higher wavelength, that is a red, right. So, this is something called shift, resulting shift, which is called bathochromic shift. The shift towards longer wavelength is called bathochromic shift or red shift, moving towards the red end of the visible spectrum. So, molecule with sufficient conjugation, typically say 5 or more conjugated double bond. What I mean by conjugated, like here you look at this conjugated single and double bond. This is typically with sufficient conjugation 5 or more conjugated double bond, move their absorption from UV region to a visible region, from more bluer region or ultraviolet region to more red or that side region. So, that is called bathochromic shift. So, molecule with sufficient conjugation moves their absorption from UV region to the visible region, that appears colored to the human light, that is why lal tomato, right, red tomato, it is red. For an so this is an example: ethylene 171, butadiene 217 and lycopene red tomato 500 nanometer, that is called bathochromic shift. Now, another important concept is a solvent polarity. How solvent polarity? I said that this affects basically UV visible spectroscopy. How this solvent, it is called solvatochroism, how that affects. So, here solvent rules, what is there, that you see this is absorbed like a chromophore that absorbs. So, chemical environment actually dictates. So, immediate chemical environment is solvent. The solvent cage can stabilize either the ground state or the excited state of the chromophore, and that leads to change of the delta E. If it is stabilizing one of those, our delta E can be changed. So, here for an example, a non-polar solvent like a hexane, for this chromophore, this is the energy gap ground state. In the case of polar solvent like water, the excited state and ground state moved apart, and that is how you have a more delta E. For pi to pi absorption, for n to pi absorption, non-polar is like this, and in polar you have changed it again n to pi star. So, in those cases what is going to happen? You shift it because delta E has increased, right. So, you shift it. So, here shifting is happening because delta is increased. So, towards blue range, that is called hypsochromic shift. So, excited state pi is generally more polar than the ground state pi, and the polar solvent like a water or ethanol stabilizes more polar excited state than the effectively by a dipole interaction, and if this is separated more, the delta has increased, h mu increase, lambda decrease. So, that is what is the hypsochromic shift. So, n to pi transition, the other transition that we looked at, these transition often involve non-bonding lone pair like in say in ketone or something. Polar or protic solvent like water can form hydrogen bond with this n orbital electron in the ground state, and that strongly stabilizes. Once it forms with a hydrogen bond with a non-bonding, it can stabilize it. So, excited state pi star is less stabilized and therefore increases delta E and causes the hypsochromic shift, or this is called blue shift. So, this is bathochromic shift red, and this is hypsochromic shift, and that is what happens. So, hypsochromic shift is happening in this n to pi transition, and here bathochromic shift happening, and therefore, now we see that depending upon the solvent our absorption is changing. Now, the third factor that basically dictates the absorption called substituent, and that is called oxochrome, right, oxochrome. So, presence of oxochrome, here say like a oxochrome, what is an oxochrome? It is a functional group attached to a chromophore, that modify the ability of chromophore to absorb the light. How it does that? By shift the lambda max or increasing the intensity, they do not absorb the significant UV visible, but shift the light. So, like if you look at here, the benzene is here, benzene is this, benzene is absorbing at 254, but if you have a phenol that absorbs at 270 to 280. So, group like lone pair electron or OH group or NH2 group or OR group or SR group or halogen, they basically can shift how much light is absorbed, and those are called oxochromes. Electron donating group or delocalization extend the pi system, that is destabilizes the excited state and it shifts basically the absorption of these lights. So, that the mechanism, the group usually acts as a electron donating group via resonance to the pi system, they extend the effective conjugation and stabilizes the excited state similar to exciting the conjugation overall, and they essentially introduce strong bathochromic shift. So, lambda max increases and a hyperchromic shift increase in the intensity. You can look at here, intensity has increased because of the molar absorptivity. So, oxochrome basically shifts the absorption or also increase the intensity. The fourth factor that affect the absorption is pH or isomerization. So, what pH does essentially? pH is crucial, right? pH introduces polarity. So, pH is crucial for containing the, like I say, ionizable oxochrome like carboxylic acid or amine or phenol, right? Here you look, the pH affects the ionization. So, methyl orange acidic form is protonated and which goes from pH 3 to pH 9, this methyl orange basic form deprotonated. So, look at the color changes essentially, color changes from say basic form at 420 to acidic form at 5010, and like here bathochromic or hypsochromic shift happens whether it is protonated or deprotonated. So, this is the one of the example, phenol which is weak chromophore versus phenyl phenoxide iron that is a strong oxophore results in large lambda max shift because of this lone pair available changes dramatically. The another factor that can affect is the temperature. Temperature variation primarily affects the vibrational and rotational energy level that can go up and down, and stability of solvent chromophore complex can be also affected by temperature. So, generally increasing of temperature leads to broader absorption band due to greater population of the excited state vibrational state, that is a hot band, and rather than a sharp peak. So, if you increase temperature, your absorption is not very sharp, basically it's get delocalized over the large wavelength. The another factor that affects is cis trans isomerization, because if you look at cis trans, electron cloud is changing. So, effects of conjugation or planarity changes and therefore, absorption changes of this, like azobenzene is one of the example where isomerization changes the absorption spectrum. So, to sum up what chromophore we have and what are basically their absorption properties. So, benzene ring, this is the structure C6H6 aromatic pi system 254, strong UV visible pi to pi transition happens, and you can have application in organic synthesis UV spectroscopy. Then carbonyl group, ketone aldehyde we have, this is the absorption N to pi or pi to pi transition, this is in biology, right, each protein has a ketonic group. The nitro group basically it is 350 to 400, strong electron withdrawing group, this is di or explosive. Azo group 400 to 500, now this is again di. The chlorophyll, the porphyrin ring, this is 430 and 600, this is large conjugated system with metal center, photosynthesis in plant, this chlorophyll is done. Heme group, again porphyrin ring 540 to 580, some metal coordination complex, oxygen transport is done by hemoglobin. Now, beta carotene, it is an example of conjugated polyadenine, its extensive conjugation absorbs blue green light, the vitamin A precursor or antioxidant. Similarly, indigo famous dye, right, dye in textile or denim industries 620. Now, methyl like a malachite green again 617, strong absorption in visible range, and it is a dye, or we use it for biological staining. So, these are common chrome 4 that we absorb. This is typical absorption wavelength, this is the key property, and this is the application. So, if you look at the application of absorption happening all the way from the basic science to the dye industry to staining of the textile or dye, and to the biology like a photosynthesis or electron transport that happens in our body, right. So, summary of the shift of terminology that we looked at. Let me summarize: bathochromic shift, red shift towards the longer wavelength; hypsochromic shift, that is blue shifts towards smaller wavelength; hyperchromic shift, increase the intensity, that means it will lead to a taller peak. The hypochromic shift, decrease in the intensity, that means a shorter peak. So, these are the shift terminology that we have discussed: bathochromic, red shift; hypsochromic, blue shift; hyperchromic, zyada, so that is increase; hyperchromic, decrease, right. These are the terminology that we. So, let us conclude this and summarize this portion. So, structure dictates the function. We looked at the intrinsic structure, conjugation length, primary determinant of the chromophores, more conjugation, longer wavelength, bathochromic shift. Environmental is the key. The surrounding media like solvent, pH, temperature significantly influence the electron gap, delta E. pH is one of the important factor. It acts as a chemical switch, like we looked at one of the example here. pH change, color change. So, this can be used as an indicator of pH. So, solvent polarity can induce red or blue shift depending upon the transition pi to pi star versus n to pi star. The oxochrome enhances the absorption. So, the attached group increases the conjugation and absorption, the hyperchromic shift, and spectroscopy terminology we looked at the precision in terms like bathochromic, hypsochromic, hyperchromic, and hypochromic. These are essential, and these are the terminology that you need to remember. Understanding these factors allow analysts to control the experimental condition, predict what is absorption is going to happen, and interpret the UV visible spectra accurately for like laboratory instrument or the application in various field: environment, biology, chemistry, dye, textile industry, forensic science, and so on and so forth. So, let us end it here, and thank you very much, and we look forward to see you in the next class with a new topic. Thank you very much.